The first keynote speaker, Professor Diwan Singh Rawat. I think most of delegates and scientists they know. He is professor and dean, recipient of several awards, and the title of his talk, A Ray of Hope for the Parkinson Disease Treatment Discovery of Clinical Candidate. The second speaker of this session, Dr. Rajdeep Roy. So he will discuss interaction, membership, executive, external relation, Royal Chemistry India Private Limited. And third speaker of this session, Dr. Satwal Singh Badsara. He is also very active scientist, and he published several excellent papers. So now I request Professor Diwan Singh Rawat to start his talk. Please.
Thank you, Professor Chauhan, for your kind word of introduction. Thanks to Ved and Professor Tiwari for their kind invitation. Actually, day after tomorrow, PM Modi is visiting Delhi University, so I had to prepone my flight, and I have a flight at 2 o'clock, 2:10 or so. And yesterday night I came to know that my talk is of 45 minutes duration. So I changed few of the slides. Now I realize that I may have 20-25 minutes. So in between I have to skip the slides. And I have a very fond memories of this place. I have been to this university, I think this is my third or fourth visit. And in one of the visits, this is for the information of the people who are coming from outside. The selection committee start at 8 o'clock here because sun rises a little bit early. And it got over around 11 o'clock, and 11 o'clock I left for airport. The flight schedule was around 3 o'clock. It started raining. Until 6 the flight did not reach. Then flight reached it, but it could not land. So I had to stay back. So stay back means one more day, and the next day also the flight was at the same time. So the flight was rebooked from Silchar, and from here to Silchar, road journey was about 10 to 11 hours. So I hope this time we will have a comfortable return flight for all of us. With this note, let me just talk about the work what we have been doing at University of Delhi. Before going to this, for the information of the students, how the organic chemistry developed in the morning, Professor Dekar talked about the urea. And as an organic chemist or the student of chemistry, we all know, the organic chemistry starts with the discovery of the urea. And Euler made it. And if you look at the history, Euler was not a chemist actually. He was a medical doctor. And he was taking subsidiary class of Professor Gamlin, and he realized his potential in chemistry. He asked him to join the lab of great men of that time who talked about the Vitalim's hypothesis. What his hypothesis was? That the organic compounds cannot be synthesized in the lab. And that fellow at the age of 28 synthesized urea in the lab and discredited the hypothesis of his own boss. From there, in 1845, another molecule was synthesized in the lab that is called acetic acid from the carbon disulfide, and that was achieved by a famous chemist of that time called Hermann Kolbe. And Hermann Kolbe was a person who made nasty comments on the discovery of tetrahedral carbon. He termed it as childish fantasy, which was discovered by Leeuwenhoek. And if you look at the chemistry, we always talk synthesis. Without structure and synthesis, chemistry is not complete. Have you ever thought who coined this term synthesis? This term synthesis was coined by Hermann Kolbe in his first synthesis of acetic acid in 1845. And if you look at, again, the history, you will find that the first synthetic drug that was in market is called chloral hydrate. It was used as a sedative, it's still used in some part of the country. So from urea till date, how much it has impacted the human life. If you look at this slide, in 1900, the average age in India at birth was 23 years, and in 2000 it gone to about 65 years. So this transition from 23 to 65 years, the chemistry and chemistry-biology interface has played a pivotal role on this, because the chemistry growth and a lot of medicines, the vaccines that came up, and that the foods, everything that increased the lifespan period of the human being on the earth. But when we talk about drug discovery, Professor Chauhan mentioned in the morning, it's a very tedious job. It takes about 15 to 18 years to develop a drug. Out of 10,000 compounds, one compound comes to the market, and it costs about 450 million US dollars. So any medicinal chemist, whoever wants to deliver a talk, this is the slide everybody would like to show you, because at the end, the audience should not ask the question, have you developed a drug or not? So this is why we show this slide. But when we talk about drug discovery, in drug discovery, actually there are 3-4 different approaches. One is called rational drug design, and there is a target-based, repurposed, and the recent one was the hybrid drug discovery. When we talk about hybrid discovery, what it does mean? Hybrid means the mix of two things, actually. This is what hybrid technically is called. So if you go to the doctor, the traditional disease is the tuberculosis. Tuberculosis cannot be treated by a single drug molecule. You need to have more than one drug. So as a chemistry student, you can think of why you take 3 tablets. Why can't we do one thing? You have a tablet A, you have a tablet B. Hook up these two tablets by a covalent bond. When you do that, you end up generating a new molecule. This molecule will have a pharmacophore from this drug and pharmacophore from this drug. If it works, it should solve the problem of drug resistance. If you look at the literature, the nature, this concept was already there in nature, but it was not exploited by the medicinal chemists. On the concept of hybrid drug, the first paper was published in biochemistry in 1997. Okay? And what is this? This is what I explained you. So the third one, sorry. Can you show me how it works? Anyone? Can you show me this, please? How it works? You can speak. No, point. Okay. Thank you. So this is the hybrid one. One molecule has 2 pharmacophores. So if that works, you can have better patient compliance, single molecule, low drug-drug interaction, better predictable pharmacokinetic properties, and all that. As I said, nature already had this. So if you look at this molecule called rifamycin B, is a drug for the treatment of tuberculosis. Okay? And this drug, this molecule is a natural product synthesized by the plants, and it has 2 different parts. In the plants, what we say that molecular biosynthesis. This part is synthesized from cyclamic pathway, and the rest of the part is from polycactide pathway. So this molecule is a hybrid of 2 different biosynthetic pathways. So when literature survey was done, it was found that all those molecules which are synthesized through more than one biosynthetic pathways have much better biological response than the molecules which are synthesized through a single biosynthetic pathway. Look at this plant. You all must have seen this plant, right? It is called Sadabaha. This plant has been a source of 70 alkaloids. Structure of 2 of the alkaloids I have shown it here, and both of these 2 alkaloids shows poor anti-cancer activity. Same plant generated 2 other molecules of the structure of those molecules like this. When these 2 molecules are hooked up by a covalent bond like this, this becomes vincristine or vinblastine, depending what you have at nitrogen, N-formyl or N-methyl. And these vincristine and vinblastine molecules became a blockbuster drug for the treatment of leukemia, FDA approved in 1963. So this concept of hybrid drug discovery was utilized by the synthetic chemists. One of the example I'm giving it here. This molecule was developed by then Ranbaxy, and it was found to be better antimalarial drug. So when we started working at University of Delhi, the first project we started working on called the tetraoxygen. If you look at the central part of the molecule, it has a 6-membered ring, 4 oxygen. So you can see it is a cyclohexene structure. And we became the first person to, we were the first who determined the crystal structure of this and showed this tetraoxygen has a perfect chair conformation as a trans. Okay? And then what we designed here, we thought to take this part of chloroquine and this part of these two old drugs used for the treatment of malaria and use these kind of hybrids. These hybrids have a 4-aminoquinoline or pyrimidine or trizine pharmacophore. So with this note, I will skip the synthesis. What we did in this case actually, we synthesized this kind of hybrid, okay? And this is the IC50 value of chloroquine, and this is the IC50 value of our compounds. So if you look at it, the IC50 value of some of these compounds are better than chloroquine. And we published in 2010. And we started this work in 2007 and 8. At that time, we do not even have NMR in our university, though I come from Delhi University. People feel that we should have everything, but we do not have anything at that time. So characterization, then getting the biology data, it took a lot of time. By that time in 2009, the concept of hybrid of aminoquinoline and trizine was published by Professor P.M.S. Chauhan from CDRI Lucknow in 2009. So the concept got published, but luckily our compound's structure was a little bit different than what these people published. So we also could publish this work. Later, the structure-activity relationship studies, what we proved, the activity improves if you go from this to this. So 3 nitrogen 5-membered ring, 3 nitrogen 6-membered ring, the activity improves. So as a medicinal chemist, PhD from CDRI, this is what we learned from there. Let's do the next step. Substitute this nitrogen by CS, then it becomes pyrimidine. And compare the activity of trizine and pyrimidine-based compound and try to do the SAR studies and draw a hypothesis that how the activity can be improved. So we made this. This is the regioisomer, these regioisomer separated. Crystal structure of the compound was determined. So this is what we synthesized, what I showed you in the previous slide. Now the difference is from this to this, 1 nitrogen is less in this compound. So next is the in vitro antimalarial activity of those compounds. If you look at it, it is 0.21 micromolar, okay? And these are 2 regioisomers. So both of the regioisomers has an identical kind of antimalarial activity. So the attachment of these pharmacophores with aminoquinoline has less effect on antimalarial activity. But if you substitute this CR by this carbocyclic amine, the activity improves by 10-fold. Next, if you substitute this by N-ethylpiperazine, the activity was up to 7 nanomolar. So if you have a nanomolar range activity, people will always ask you, what about the in vivo activity? We did the in vivo activity of those compounds. What we found that 30 milligram per kg body weight for 3 days is 80% cure. So this work was published in 2012 in ACS Medicinal Chemistry. So once you have nanomolar range compound, you would like to, optimize the lead molecule. So this is what it was done. So we started from 2.45 micromolar, ended up to 7 nanomolar, good in vivo activity. So we made all these kind of molecules. So I will not go to talk about all of them, but I will talk only 1 or 2 cases. So the next goal, what we thought, to substitute this linker by a rigid one like this, piperazine. These 2 series. Second series was keep piperazine in between, but introduce the flexibility also. Let's see what happens. So it was like this. So if you have a rigidity in between two pharmacophores, activity drops. But if you have a piperazine there and the flexibility is there, the compound is much more active, good solubility. It was 50 times better than chloroquine. It has very good in vivo activity, 100% cure. I will skip this. Okay, this is a very good example for a student's purpose. You all know the structure of paracetamol, right? And you all know the case of Dolo, right? During this COVID, we started taking Dolo. Dolo is nothing but paracetamol. What is the difference between the paracetamol and the Dolo? The difference is the dose level, 650 mg. How the 500 mg of paracetamol was established? It depends on the therapeutic index. So this company just increased the dose level and sold it to across the country and made huge amount of money. But paracetamol has liver toxicity. You must know that. So what happened? This drug called amodiaquine was discovered, sold in the clinical market as a drug for the treatment of malaria, better than chloroquine, but later on it was withdrawn. As I mentioned in one of the slides, it cost about $450 million. So if you develop a drug and then take it back, you can understand the fate of the company. Why it was withdrawn? Because of this metabolite. This metabolite is analogous to the metabolite generated by paracetamol in our liver. This metabolite is toxic. So if your liver is infected with any kind of disease, you should not take paracetamol because it can be fatal. So what we did in this case actually, we substituted this OH group by fluoro, and this N-diethyl group from our pharmacophore, this one, what we showed you the previous. So this is the structural difference between the drug molecule. This portion is substituted by this. So what's the activity level? 100% cure. Much better T-half than the amodiaquine. So conceptually, we proved that if you substitute the groups, you can avoid the toxicity of the molecule. The next one is now, look at this, aromatic ring. This is cyclohexyldiamine. What is the activity now? 2.2 nanomolar. Not only that, in vivo, it cures, and after 60 days, the mice weight increase. So complete parasitemia clearance. At this particular time, actually, we entered to a collaboration with Professor Kim at Harvard Medical School. The reason was, when he read this paper, he showed his interest that he wants to test these compounds for Parkinson model. And the motivation of doing so was this paper published in Nature in 1987 by Sanders. And conclusion of this paper was that chloroquine can retard the progression of Parkinson's disease. But unfortunately, there was no follow-up of this work. What Professor Kim did actually, Professor Kim took about 960 FDA approved drugs, screened all those drugs against the target what he's developed, and found 3 hits. All these 3 hits, if you see, has 4 aminoquinoline pharmacophore. One is chloroquine and another is amodiaquine. And our compound has structural similarity with this. Much better than chloroquine in malaria model. So let me just tell you what this Parkinson is. We all know it is a neurodegenerative disease. I don't know how many of the students knows how many neurons we have in our brain. Our brain has more than 86 billion neurons. None of the neurons makes a physical contact to each other to pass the message. This is called a synapse. This neuron, this is called a synapse. And the information from this neuron to this neuron is transmitted from the ions. So now you can understand how many synapses will be there. So this Parkinson disease is caused due to the deficiency of dopamine, and we have a dopamine neuron in the brain. And in the part of substantia nigra and seritum, this is the part of the seritum. And this is where the dopamine plays a role. So when dopamine neurons death takes place, concentration of dopamine decreases, you will have a Parkinson's symptom. Once that happens, there is another protein called alpha-synuclein. Misfolding of alpha-synuclein takes place. The lumps of alpha-synuclein accumulates this region. That aggravates the disease. There is no drug. L-DOPA and its derivatives are given to manage the disease. It cannot cure. In a nutshell, this is the normal, healthy human brain. This is the dopamine neuron, and it generates the dopamine, and these are the alpha-synuclein protein. Perfect, no problem. But once this gets disturbed, you don't have a dopamine here, and this protein forms these kind of lumps. You have Parkinson now. All you need to do is stop this process. Either this or this. You can have a drug. But that's not that easy, as I said before. There is a target which people thought it is an orphan target, NER1 enzyme. Orphan target means that the enzyme that does not have a binding pocket. All these years, the scientists felt that this NER1 doesn't have a binding pocket. But Professor Kim's work proved that it has a binding pocket, the NER1, but the binding pocket is very tight. There is a mystery molecule, this is what it is taught, which binds with the NER1, and NER1 get activates. Once NER1 get activates, it stops the death of the dopamine neuron. So जब आपकी dopamine neuron की death खत्म हो जाएगी, dopamine बढ़ेगा. Dopamine बढ़ेगा, Parkinson का problem खत्म होगा. Next one is this. If you can stop the aggregation of alpha-synuclein, that can also help. So with this, actually we started collaborating in 2012. First lot we sent about 120 compounds. We got 4 hits out of that. And it was funded later on by MJ Fox Foundation. We screened about 650 new molecules. Multi-step synthesis, as I mentioned, we have a regioisomers. So purification is really tedious. So if you look at it, the EC50 value of chloroquine, which nature says it can retard the progression of Parkinson's disease, was 50 micromolar. And our compound's EC50 is as low as 0.5 nanomolar. Now this molecule, this one, how it was, no, actually what happened actually, we published this paper in Royal Society Advances for malaria. Then our collaborator wanted to have this compound in more quantity so that we can do more biology work. I asked my student, this person joined from Hyderabad, to make this compound. He made it, but he wanted to make it 100 milligram scale. During that period, he isolated one polar compound that was 8 milligram, which most of the students will miss out. 8 milligram. Who cares? If that compound is active, your life will be miserable. And this is exactly what happened in this case. So when we got this compound, I felt this compound has a better potential than the compound what we have earlier, and found the same thing. This compound was 0.5 nanomolar active in Parkinson model. So it means what? It means you have to make this one more now for in vivo activity. We improvised the synthesis, did that. So batteries of compound we had with a very low nanomolar activity, again Parkinson model. Now just show you how it works. So this is the luciferase enzyme using NER1 ligand bind domain. This is the chloroquine dose. This is the dose of our compound. Chloroquine dose is 100 micromolar. Our is 20. So 5 times better than chloroquine. This is the using full length NER1. This is the ligand bind domain. This is the full length protein. Same result. This red dots, this is the vehicle, normal cell line. So the red dots mean the dopamine neurons. So when this treated with MPTP, a compound that causes a Parkinson disease, you can see the red dots gone now, decreased. When this is treated with chloroquine, it started regaining higher dose. If you look at this, our compound, red dots are much more than chloroquine and even this one. So what we proved here, it doesn't even stop the death of the dopamine neuron, it increased the dopamine neuron in brain. So this is the compound that causes the Parkinson disease. So this is the serotonin part of a healthy mice. When this mice is injected with this compound, Parkinson progresses. This is how the serotonin part looks like now. When this is treated with L-DOPA, you can see it, not much different. But when this is treated with, this has a low battery. When this is treated with our compound, if you look at this figure with this figure, it looks identical. It cures. Not only that, it stops the alpha-synuclein aggregation, this figure to this figure, identical. The beautiful mechanism, these are the two mechanisms what we proved. It stops the death of dopamine neuron, number one. Number two, it stops the aggregation of alpha-synuclein. Number three, it induces autophagy. Autophagy, what it is, do you know? Autophagy में 2015 का Nobel Prize मिला है एक Japanese professor को. What it says, in nutshell, if you talk about autophagy, autophagy means if your cells are under stress, to survive, those cells eat up the cellular garbage. Cellular garbage means the dead cells or cells that causes the diseases. This is how the system get cleaned. So this is why maybe our saints, all that, used to have a more fasting, longer life. So this is what the concept is. So what our molecule is doing, it is inducing autophagy. What is that? So this is the normal starving phase of the cell. When this is treated with this compound, which stops the autophagy, all red dots are gone. When it is treated with chloroquine, little bit of red dots comes back, but it is much less. But when it is treated with our compound, all red dots comes back. So it clears this cellular garbage also. So where are we now? We started in 2012 on this project. Initial identification, synthesis, preclinical candidate, 20 candidates what we have. Now 2 candidates are selected for phase 1 human clinical trials, and this is the step which need to be taken care of. And this is the mice injected with Parkinson. After treatment, at the end, the mice survived and it was much healthier. And this work has been accepted for publication in Nature Communication very recently. So these are the people who made this work possible. All of them are well settled now. And the ultimate happiness for any medicinal chemist is if your molecule is taken up by industry. So this is the industry who take up our molecule, we transfer the technology. These are the 3 molecules which they listed on their website as pipeline molecules. Just last month, the Hanall Biopharma and Dowing Pharmaceutical Industries entered in agreement with Nervan to develop these molecules for the human clinical trial, and it says in 2013 they will start the human clinical trials. And this human clinical trial has been funded by M.J. Fox Foundation, who supports the work related to Parkinson from US. Because as you know, the work was 2012 में जाके 2013 में पेपर आया, कोई PhD student रुकेगा नहीं आपके. So we started working on some catalysis work, methodology work. We made a huge success there also, many cover page. Many of the papers have been highlighted by Synfact. Recently we got one JOC, and the work was possible because of funding from these funding agencies. More of the malaria work was collaborative with Savannah Khan. As I said, Parkinson was with Kwang Soo Kim from Harvard Medical School. These are the students who made this possible. And this is my current batch of student. Thank you very much for your patience here.
Thank you, sir. Are there any questions? Let us open the floor. If you have any query, okay, please.
You mentioned that linker, in one of the slides, you mentioned that linker plays a role in activity. So I'm curious to know how the flexibility of the linker present in that particular drug molecule can change the activity drastically. What would be the possible mechanism?
As I said, because in case of malaria, because our target was heme, okay? Heme is not a protein, okay? It is a kind of association, pi stacking, okay? But the other part is the PFCRT inhibitor, okay? So that might be inhibiting the PFCRT enzyme, number one. But in case of Parkinson, what we found actually, as I said, the binding pocket is too rigid. So small changes leads to the drastic change in the activity. Okay? But in this particular case also, if you have a rigid molecule, as I said, if you hook up these two by piperazine, becomes very big molecule now. So it may not fit. But if you have a flexibility, then there is a chance that it can go and fit in there. So that may be the reason.
One more? Anybody? Yeah, please. Please, can you, there's one more.
Sir, thank you for your very informative talk. So I want to know what are the factors which affect the concentration of dopamine which leads to Parkinson's disease?
I didn't get your question actually.
What are the factors? Factor, then may be the external or internal.
Okay, so one of the factors what we call the environmental factor.
Oh, yeah.
Okay, so there are many environmental toxins. One of the toxins what I showed you, the compound, that is the environmental toxin actually.
Okay.
And another one is the genetic.
Yeah.
The alpha-synuclein portion what I talked about, it is the genetic one. And MPTP what I talked to you is the environmental.
Thank you, sir.
Okay. Thank you so much. Sir is in a hurry. He has to go back. He has to catch a flight, it seems. So thank you so much, Professor Diwan Singh Rawat. But before you leave, here's a memento I would like to ask sir to give. Sir, please. Thank you so much. Couple, come on. I'll call the next one. Okay, okay. Next, I would like to call upon Dr. Satpal Singh Pansara, Department of Chemistry, University of Rajasthan.
Wait.
Huh?
As the first he will finish, then he has to go.
Oh, is it? I'm so sorry. I'm so sorry. Interaction with Dr. Radip Roy is a membership executive, external relationship, Royal Chemistry India Private Limited. May I call upon Dr. Pradip Roy? Yeah, please. Please take the stage.
This is the pointer.
Good afternoon, everybody. So, first of all, I should say that a huge thank you for the organizers for organizing such a beautiful event over here. And I want to thank you all the delegates, the distinguished speakers over here, especially Professor Singh, and eventually couple of my friends and seniors happens to be the faculty members of industrial chemistry as well. So it's a good reunion for me as well, being here in this beautiful city of this northeast part of the country. So first of all, I should say that a huge apologies from my side because again, thank you to Professor Singh to prioritize my talk. Sorry about that because I have a flight right after this one. So I will take maybe 10-15 minutes and then I will conclude actually. So obviously just after this beautiful scientific talk of Professor Rawat from Delhi University, it will be a totally non-scientific talk in that sense, but we are also associated with the chemistry that we are all discussing about. Chemistry is everywhere, and I work as a membership executive of the Royal Society of Chemistry India office in Bangalore. So let me just give you a brief idea that what we did and what is our membership benefits that we have, mostly for the young researchers and the students. So, as you know that we are, everybody knows that RSC originated from UK, but right now it's almost worldwide and we are globally located actually. So I'm not going into the details of it. Apart from all other countries, we also have an office here in India, and that office located in Bangalore, but we have a very small group of like 13 employees. So being such a big organization, in India, our activity, we are trying our level best with a very minimum amount of employees. So we are based in Bangalore, as I have mentioned, since 2020. And we have in India right now over 2,000 RSC members, and out of those 2,000 RSC members, almost 400 are of them from the senior level, that is FRSCs, and obviously rest of the members are from mid-senior level to the young researchers. And according to our Indian activity, we are very closely working with different chemical societies like CRSI, Indian Chemical Societies, etc. And I'm giving you a brief idea that what we are doing right now here in India, what is our activity. So mostly we are doing this, we are inspiring young researchers in STEM, and there is, as we all know, that there is always a huge dropout or a very low percentile of women scientists in chemistries are still quite obvious right now. So we are doing a survey on this, that breaking of barriers of India, like what is the gender diversity in the area of chemical sciences, as well as we have, apart from these different survey, we have done a lot of educational kind of program, and one of them is Yusuf Hamid Inspirational Program, mostly for the school childrens and to inspire teachers as well, both college teachers as well as school teachers. And apart from all these, we have in UK India Innovations Program as well, and RAC CSR Jigyasha Partnership. Now, just giving you a brief idea regarding this CRSI partnership, that we have signed a cooperation with CRSI since 2007, and it has been renewed, I think, each and every after 5 years. And very recently, the last CRSI that we had in JNU New Delhi, we have renewed the cooperation with CRSI again for another more 5 years. And the good thing is that I just want to announce you that very recently we have decided that those who are CRSI members but has not received any RAC membership yet, those will be provided a 1-year complimentary or rather free affiliate membership, and obviously according to their experience later they can upgrade their membership from affiliate to associate member or even FRSCs as well. So this is one of the key vital decision that we have made in that MOU, and we have each and every alternate year our CRSI RSC program in UK as well, where we usually invite lot of Indian chemists from India who are associated with CRSI, and they travel to UK and they speak on the current activities of chemistry, mostly in the sustainability, as we have mentioned, which is very important for this conference in those areas. So this is some of the glimpses of the last CRSI that we have. Very recently we will going to have another CRSI, that is a CRSI ACS in NIT Rourkela. So I'm not going into the details of that. So just to give you a brief idea what we usually do, we all know that we publish journals, but not only journals, apart from journals we do have a lot of books, databases like Mark Index, ChemSpider, and this learned chemistry. These are all the databases that we have. And apart from that, there are quite a few, pictures or flyers of different journals as well. And we have a regular, magazine which is called Chemistry World. So these are the facilities. Once you will have your membership, we will have these regular updates regarding these journals and all those things that I will come into the details. So just to give you a brief idea that as the senior professors, they have mentioned in the inaugural session that science has no boundaries and chemistry is everywhere right now. It's very much diversified. A lot of branches have come out. So this particular slide shows that lot of very specific areas of journals has been introduced, not only from the Royal Society of Chemistry but from the other publishers as well, and we are also one of them. So just to give you a brief idea, so I have a request to the young researchers: do submit your research to Royal Society of Chemistry. Just submit your good research and we will be happy to accept your research. Means I'm not the person to accept that one, but obviously there will be a review process and everything will be going on just to make sure the quality of the research work. But we encourage to publish your good work or to have your good science in RSC journals. So these are some of the flyers that I have mentioned over here, and these are again some of them are shown over here as well. And I just want to mention that RSC has a very recent specific journal on sustainability as well, that you can also submit your sustainability related work in that particular journal called RSC Sustainability. So right after this, I should thank our reviewers, not only from India, from all over the world, because they are the main stalwarts, or rather I should say the stakeholders who will ensure the quality of the research paper. And we do have a lot of them from India as well, like 22. We thank all these associate editors, the editorial board members, and as members, all the senior members who are like 2000 FRSs were engaged. We have been engaged reviewing our research journals. So now right now I'll give a little bit brief idea regarding our memberships as well. Those are the publishing things. Now regarding the membership, I just want to tell you, mainly to the young researchers, that we do have different categories of membership. So from the left side to the right, on going from left to the right, it follows the seniority of the researcher. So obviously affiliate or student member for the young researchers, those who are doing PhD, associate members, obviously those who have a primary degree like PhD or something like that, they can be applicable for this associate membership. Members who have their first degree, rather the PhD degree, with a couple of years of experience like postdoctoral experience, and college or university young researchers or early career researchers can be applicable for the members. That is called the MRSC, and obviously we have the senior members called FRSC as well. Apart from all this, we do have lot of registered tag for our chemist as well, starting for RCI tag to RCI and chartered status. Just to give you a brief idea, after having your membership, what we will have. So we do have a lot of social events. You will be able to know and you will be able to participate in a reduced registration fees with different social events that we have. You can be a member of different networks. Whenever you will apply for the membership, you can choose the different networks that we have according to your preference in which area you are working. Suppose if you are a protein chemist, you can choose your networks in protein chemistry as well. If you are a crystallographer, then you can choose a networks for crystallography, where you will meet with the people with a similar interests, and you can share your knowledge as well. So you will be able to invite a lot of webinars called scientific lectures. So those email alerts will be continuously coming to your email ID, and you will be able to receive those all these alerts each and every time from our side. And there will be like well-being seminars and ChemSocs as well. What is being well-being seminars? Those who have early career researchers but don't know how to proceed in future, or what are the different opportunities that you have. In that sense, we have a career guidance with an expert team where you can set up a meeting with them and they will help you, just to give you a good idea that what area or what way you should proceed in your future. And KEMSOC is nothing but a student society. I would always encourage to a particular university or institute to form a student society. We do have this facility where we sponsor some amount of money annually, where you can event a small program like a popular lecture with some snacks or something like that. I am not saying that the budgetary amount is very much high, but this is all about the students. Maybe 30, 40 or 50 students can come together and they can make a KEMSOX or Student Chemical Society, and they will be able to get regular funding from RSC as well for organizing different chemical science related seminars. So I'm not going into the details of these professional communities that I have already mentioned. There are 9 divisions, and when you are applying for the membership, all these options will come, and accordingly you can choose your preferences. And as I have mentioned regarding the knowledge, I should say that there is always a opportunity to have the access of RSC's virtual library, where you have ample books on different topics that you can choose. And apart from that, there are lot of, chemistry world, there is a magazine called Chemistry World, from where you will get regular update, where the current scenario of the chemical sciences and the hot topics, what is going on surrounding us, will be notified in daily basis. And we do have another magazine called Voice as well. And obviously at the same time, you will have an access of a particular journal of your choice, where you will have unlimited views, but a specific amount of downloads. For students, it can be, I think, up to 10 downloads. And according to the seniority of the membership, you will have 20, 30 something downloads. Suppose if you are a sustainable, you are working on green chemistry, then you can choose a journal on green chemistry or sustainability, and you will be able to access that one in your, in any internet access that you have, even from your home as well. So as I have mentioned, that we do have a career guidance group, the well-being seminars that I have mentioned. We do have a specific group of experts who will encourage you to, give you the right path to choose for your future. And this slide is very much important, the grants and funding, what people always, are looking for. So we do have 4 different kind of grants. Scientific meeting grants, where you can go to you can organize a scientific meeting, and for that up to 2,000 pound can be sponsored. And at the same time, there is a huge opportunity for the second one over here. This is for attending conferences abroad. It's not necessarily any RSC-led program. It can be anything like Gordon Research Conference or whatever. You will be able to get up to 800, but mostly right now we are giving 500 at least. That means 50,000 at least. You will get the to and fro airfare kind of a support from us. And research fund, this is very much important. Those who have joined to an institute, like very early career researcher or something like that. There is a different meaning of early career researchers in Western countries and in our countries. So I should mention this one for the assistant professors, those who have joined and want to do good research. They can get up to 5,000. It has been increased to 5,000. That means almost 5 lakhs INR. And there is another option called researcher mobility grants for PhD students and early careers. Obviously, that should be within 10 years of your PhD. You can get a grant up to 7,500, where you can go to a different institute or organization and do a collaborative research. But for avail all this facility, you at least have to have a membership with RSC, and that obviously there is no barrier of different category of membership. For all members, these facilities will be available. Sorry. And there is a grants for carers, which is in that sense not that common here in India, but it is internationally acceptable. That means those who have, this is mainly for the women, those who have babies and they have a career gap or something like that. For them, we do have grants to, attending a conference or going for a different organization for research as well, and up to 10,000 per year for 5 consecutive years can be sponsored. And there is another one called assistant grants, which is also not that much familiar here in India. Those who have physical disability or, especially I should say active person or something like that, for them there will be another same kind of facilities available for 5 consecutive years, £1,000 a year. That means almost 1 lakh. And this is the current subscription fees that going on in India. All these rates are after 50% reduction. But what I'm trying to say, suppose for member, if it is 75, that means the original rate is 150. So for India, it's 50%, reduction or reduced rate is going on. So obviously all these are in GBP pound. So you have to multiply it in around 100. So for the students, you have to pay 1,000 rupees a year for this affiliate membership. I should encourage students to go for the affiliate membership, mostly for the PhD students, where you will have this the benefit that I have mentioned. And another last comment I just want to make before finishing this one. Very recently I have visited to NEIST Jorhat, with NIST Jorhat, a CSR institute I think many of us may know. And we have decided from NIST, almost 70 students have registered for the membership and they have formed a KEMSOC. And we thought that we should look at the Northeast, and why don't we consider Mizoram University as well. That's the reason I traveled far from south to here, just to encourage you that you can also make a group of students with a affiliate membership, and we are planning to make a local section. I'm coming to the local section of Northeast, for Northeast, dedicatedly for Northeast, because right now the different local section is mentioned over here. For the Northeast members, they come under the East section, East India category. But if you make, if we could be able to make a new local section over here, then a new local section will get a specific funding or a budgetary funding for each and every financial year, and that funding can be used for different purposes or different programs, as well as for different grants that I have mentioned. So formation of a new local chapters here in Northeast will be a high priority for us, and we seek help from you as well, obviously, because we should be able to convince to the UK team that there is a considerable number of members are really interested to join our community, and we could go ahead with this. And we have already proposed this one to our UK team. We are going to make two new local chapters in our proposal. One is this Northeast, and another will be for Central, mostly for the Madhya Pradesh, Chhattisgarh area, because recently the IISER Bhopal and a lot of people from IIT Indore, they are also very much interested, because it comes under the North India category. So we thought that why don't we go for two different local chapters from Central and Northeast. So we could think over it, and if we could make it, then it will be a very good opportunity for us, and there will be a lot of budgetary fundings will be available. So that's it. I will not take time. Thank you. If you have any question, you can ask me right now, obviously. And just want to make a quick comment at last, that I will write to you after the program regarding the membership related things, because it will not be possible to understand very quickly what I have said right now. So you will be able to, hopefully you will be received one email from our site. And as well as I just want to make one announcement right now, that those who will win the poster prizes, those students or the research scholars will get a 1-year complimentary affiliate membership from our side as a prize, awards. Apart from that prize money that we have, I think Professor Singh will be the best person to convey this one. So I just request the organizers to share their email IDs right after the program for the poster prizes winner, and they will be awarded with the 1-year complimentary affiliate membership. Thank you. Thank you, everybody. Thank you.
Any quick queries?
Is there any scope of live membership?
Yeah, so the live membership scope is something like that. If you have a continuous number of renewal of your membership for 8 to 10 years, then automatically it will be converted to live membership, but it is not from the very beginning right now. Yeah. Yes.
Any other?
So for the FRSs is 87 right now. So as I have noticed that those who are FRSs since for the last, I think, more than 8 to 10 years, now they have converted to live members, and they didn't know
no need to pay. Yeah. Thank you.
One short query, any? Okay, thank you so much, Dr. Rajdeep Roy, and we'll take note of that local section notice, especially that one, and the others which you've mentioned also.
Yeah.
So I would like to call upon to give the gift to.
Thank you.
Okay, thank you so much. Thank you so much. He's also in a hurry. They need to catch a flight with the former keynote lecture. And now I would like to call upon Dr. Satpal Singh, Bansar, Department of Chemistry, University of Rajasthan, to give the invited talk.
Yeah, very good morning to everyone. At the outset, I must thank the organizers, in particular Professor Ved Prakash Singh, for providing me this platform to discuss some part of research work which we have carried out at University of Rajasthan, Jaipur. So before I move to my technical details, I bow my head to my guru, Professor Basavaya, for shaping me a organic chemist, and I am enjoying the glory of his training and blessing and support till today, and I hope it will be continued for forever. So at University of Rajasthan, basically my research group is involving in the development of novel organic methods via CS functionalization, cross-coupling reactions. No doubt we are also doing our family business, that is Bellisman chemistry. And very recently we have started working on a very hot topic, that is electroorganic synthesis. And by using all these things, we are just trying to contribute by developing green and sustainable protocols for carbon-carbon, carbon-sulfur, carbon-selenium, carbon-phosphorus bond formations. So basically, my talk will be divided in two parts. In few slides, I will be just discussing the traditional methods, you can say metal-free organic transformation, and the second part I will be discussing the electroorganic synthetic part. So we have mainly contributed in carbon-sulfur bond forming reactions, and those developments were summarized in form of different reviews in collaboration with my good friend and my postdoc supervisor, Professor Lee. And these are the cover pages of those review articles. So when I joined University of Rajasthan in 2014, I started working, I got laboratory space and all these things in 2015. And it was a discussion that how to work at a place where nothing is available, there is no infrastructure and no instruments. But since the training which we obtain from our supervisors and our mentors, it's always important to work very hard to contribute in the way which you can do. So I started working in Bellisman chemistry in 2015, and we could successfully employ a variety of Bellisman adducts for regio and stereoselective carbon-sulfur coupling reaction with variety of sulfur surrogate, which we published in Tetra and RSA Advance. Also, a catalyst-free and substrate-switch dual functionalization of Bellisman adducts were also reported by our research group in 2019. And we have also developed a catalyst-free diastereoselective CP bond formation between these Bellisman alcohols obtained from isatine with the different phosphorus surrogate like phosphine oxide and phosphides. We have also developed a highly atom economic catalyst-free and solvent-free phosphorylation of chalcogenides. You can see that when we use, we mix this diphenyl phosphine oxides with disulfide or diselenide or ditelluride even, just on a silica gel and put at room temperature for just 6 hours in open flask, we could obtain these corresponding products in good to excellent yield. And believe me or not, these kind of transformations were earlier reported in very expensive transit metal catalysis. We have also developed a iodine catalyzed process for the regioselective aryl selenation of alanines using diselenide as selenium source. And you can see here that the reaction was stereoselective. The regioselective mean that if we have a ortho position blocked, then could obtain the resulting selenization at para position. And in case of when we have blocked the para position, the selenation occurred at ortho positions. We have developed a atom economical catalyst-free hydrosulfonation of densely functional alkenes. This is the alkenes, basically it's a Bellissman alcohols obtained from astatine and methyl acrylates. And you can see that it's very simple reaction. I love this reaction very much. Just you mix this sulfonic acid and this Bellissman alcohol in water and ethanol solvent system at room temperature after 2 hours in general, we get 100% conversion of the starting materials. And there is no column chromatography was required, just evaporate the solvent and crystallize it. We can get fine crystal of these oxindole containing sulfones in 72 to 86% yield. And it's very important synthetic protocol, which is where we can have 100% atom economy, complete diastereoselective protocol, and no doubt reaction process at room temperature. And as you can see that mostly the water and ethanol is considered as a green solvent, so we can say it's a green and sustainable protocol. Coming to the electronic synthesis for student point of view, I hope you all will be knowing the Colway synthesis, like we can perform the organic synthesis under the influence of electricity. So due to lack of time, I'm not going for introductory part of this electronic synthesis. I'll directly moving to my synthetic part of this work. So what we did, we just developed a electrochemical protocol for the dual functionalization of these alkenes, that again these are Bellissman alcohols, which I shown in last slide, and treated with thiol and cinolone using carbon as anode and carbon as cathode in acetonitrous solvent and lithium perchlorate as a electrolyte at 10 milliampere in a undivided cell. We could obtain the corresponding oxindole containing thioether and selenoethers in good to excellent yields. These molecules are highly important. I'm not going to discuss the biological importance of these molecules, as you all might be aware. That's oxindoles oxygens are highly bioactive compounds, and when we inject or we connect with sulfur molecules, these sulfur containing molecules also also contribute significantly in terms of biological relevance. So next is we have developed a electrochemical method for thiamical and thioacetylation process in one pot process. So we can see that when we check the literature, this kind of thioacetylation process were known in the literature using transition metal catalysts or by photocatalyst, and we could not find, best of my knowledge, a process where we can perform both the things in one pot operation. So we thought that what if we try this reaction in our electrochemical condition, and accordingly we tried the reaction of this cyclohexanone 1A with benzene thiolene 2A using carbon electrodes, and you can see that a variety of electrolytes were screened in different solvent system, and you can see that lithium perchlorate in this case in DC diethyl solvent system after 4 hours provided the 88% isolated yield of this interesting trisulfane molecules. So once we have optimized reaction condition in our hand, we study the substrates of these cyclic enones with aliphatic thiols, and you can see here that a variety of aliphatic thiols underwent this electrochemical thiamical and thiacetylation process to provide the corresponding adducts in good to excellent yields. Not only simple cyclic enone, but also substituted cyclic enones were found suitable for this electrochemical carbon sulfur bond forming reactions. No doubt, when we block this beta position with methyl group, this substrate was not found suitable for this electrochemical carbon sulfur bond forming reactions. Further, aromatic thiols were also found competent for this these electrochemical reaction conditions, which can be evident by this slide, where you can see that a variety of thiols possessing both electron donating and electron withdrawing group reacted well with different cyclic enones, providing the corresponding trisulfides in reasonably good yields. Further, to find out, to study that which process is happening first, either it is thiamical or thiacetylation, we perform a control experiments where we use when we treated 1 millimole of this cyclic enone with 1.2 equivalent of benzene thiol, and which will resulted in the formation of this thiamical product 3A- in 90% isolated yield, which clearly indicates that this process is going through first thiamical addition followed by the thiacetylation process. And using this strategy, we synthesize a variety of interesting hybrid molecule. You can see where we can treat first this cyclic enone with one thiol, and after the thiamical happen, we can add a different thiol, and which will result in the formation of these hybrid molecules. To find out a plausible mechanism, we performed control experiments, and you can see here that in presence of tempo, that is radical squenger, where the yield of 3A was reduced to 10%, and when we enhance the amount of tempo from 1 equivalent to 4 equivalent, there was complete inhibition of this reaction, which clearly suggests that the reaction is going through a radical pathway. No doubt we could isolate this species 6, where we treated this thiol with tempo, which is also clearly indicate that this method, this procedure, electrochemical procedure is going through a radical pathway. We have also performed CV experiment. I am not going into detail. So on the basis of these control experiments and CV experiment, we could propose a plausible reaction pathway, which you can see over here, that initially this thiol can get oxidized at anode to generate this cation radical, and which on removal of H plus generate this null radical, which will take in a Michael fashion, followed by there will be a take of other molecule, and at end of the there we can have the this final product in our hand at cathode. Next is we could develop a electrochemical synthetic protocol for the synthesis of bis-indolyl methane. Bis molecules are highly important because it contains 2 indole moieties, and through via the bis-aerylation of carbonyl compounds. So when we check the literature again, you can see that number of reports were available for the synthesis of this molecule, which clearly indicates the importance of this molecule, in particular of biology. So we thought that let us try this reaction and under electrochemical reaction conditions, and for that we chose this benzaldehyde and indole, simple indole 2A as a model substrate, and charged them in a undivided cell using carbon as a anode and carbon as a cathode at 10 milliampere, and we screened a variety of electrolytes and solvent system, and you can see that when the reaction were charged in lithium perchlorate in acetonite solvent, just after 90 minutes, we could obtain the corresponding beams 3A in 84% isolated yields. To study the substrate scope, a variety of aldehydes, aromatic aldehydes possessing different substituents at ortho, para, meta, having electron donating group and electron withdrawing group, all were well tolerated under these electrochemical reaction conditions to afford the corresponding beams in good to excellent yields. Not only aromatic aldehyde, but also heteroaromatic aldehydes and ethyl glycosate, as well as alkyl aldehydes were found compatible for arylation with indoles under these electrochemical reaction conditions. So all the compounds were well characterized by spectroscope data and mass data analysis. Further, we extended this method for the ketones, and I must accept this that the simple ketones were not found suitable for this biserialization process. We got a mixture of product. However, when we perform the reaction with E13 derivatives and diketo compounds, we could obtain the corresponding adducts in good to excellent yields. This work with this last year published in JOC. Again, these are the control experiment which clearly indicate a radical pathway, and we could find out this coupled product 6 in HRMS data analysis. This is the CV diagram just for a student that by CV we could just try to find out that there is any interaction between both these species or not. Maybe there may be formation of some intermediates which will, due to that the oxidation potential of this substrate will differ, and by this as well as by control experiment we were able to propose the plausible reaction mechanism, which you can see here that initially at anode this indole moiety can get oxidized to form this cation radical, and again there will be removal of H plus, and this the radical will generated. This will react with one of the LDN molecules, and this intermediate will form, followed by different steps. You can get the resulting beams in this reaction media. Very recently, we have also developed an electrochemical protocol for the regio-selective and stereoselective carbon-sulfur bond formation. You can see this is the Belli-Silman iodide, we call it allyl iodide. So, the interesting part is that when we have ester group as an electron withdrawing group, it has Z stereochemistry, where we have a nitrile group, it has E stereochemistry across the double bond. So, we wanted to enjoy the glory of this interesting stereochemical differences for carbon sulfur bond forming reactions, and for under our conditions, and you can see that we started the reaction with this iodide 1A and treated with disulfide 2A, again in carbon electrodes and using different electrolytes and solvent systems, and we were lucky to obtain this product 3A in 78% yield when we performed the reaction using lithium perchlorate as a electrolyte at 25 milliampere constant current using DC and estonityl as solvent system. And data analysis of this product revealed that there was 100% retention across the double bond. You can say that when we start with Z, we could obtain the product with Z stereochemistry, as well as this kind of reaction generally goes through a radical method. So there is a possibility of isomerization from the radical from here to here. So that is benzylic position. So, but the proton and NMR crude mixture shows that there was no formation of other isomer, that is carbon sulfur bond formation at benzyl position could not occur. Therefore, I can say that reaction is completely regioselective too. To study a substrate scope of different alkyl iodides possessing substitution at different position having electron donating as well electron withdrawing group underwent for this electrochemical carbon sulfur bond formation with both aryl as well as alkyl disulfide to generate the corresponding allyl thioether 3. And we also checked the compatibility of this reaction with thiols, and the results are shown here that when we have this hetero aromatic thiols and treated with this iodides, we could obtain the corresponding thioethers, allyl thioethers in almost 70 and 65% yield with complete with 100% stereoselectivity and regioselectivity. As I told you that when we used the allyl iodide for that was having E stereochemistry again and treated with with different disulfide under optimized reaction condition. In this case also there was complete retention in the stereochemistry across the double bond, and in this case also we could not obtain the other isomer, which clearly suggests that the reaction was regioselective in nature. For the first time, we have synthesized allyl thioethers when we reacted, because the problem of these such kind of compounds is purification and stability. But we were fortunate enough to isolate these compounds, these allyl selenoethers using column chromatography, and the results are shown over here. Again, control experiments were performed by using, radical sequencer experiments, and you can see over here that when we perform the reaction in, I just try to, I forgot to mention that all the reactions were performed in current off situation, but none of reaction proceeded without current. So that clearly indicates on all the electrochemical procedures I shown that they proceed through under the influence of electricity only. So as again, when we perform this radical trapping experiment using galvanized radical, we could not obtain the corresponding LL ethers. Also, we were lucky to obtain to see the this intermediate 9 in HMS analysis, which clearly indicate that this reaction also proceeding through a radical mechanism. And you can see that as you may ask me that. See, in last slides I shown disulfide getting oxidized, but here I am showing that disulfide is getting reduced. So it is known in literature well that the disulfide can have oxidation, can get oxidized as well, can get reduced. So here in this case, first disulfide got reduced at cathode and to generate these two species, and this species will react with the radical which formed at LL iodide when LL iodide underwent reduction to generate this iodide negative, and this radical formed, and the coupling of this radical with this radical will generate the final product. And the formation of iodine we confirmed by the simple starch test, when we just added few drops of starch, few drops of this reaction mixture in the starch solution, that which got turned in blue, which clearly indicate, which clearly supported our this reaction pathway. Very recently, we have developed an electrochemical protocol for the direct CH sulfinylation and selenization of chromone fused endogenous skeleton under electrochemical condition for the first time. So these are the endogenous moiety having chromone chromone framework over here. So why, see, one can ask me why I am using these molecules and why I have to functionalize these molecules. As I told you that I am not going the biological relevance of these molecules. These whatever molecules I am showing over here have been proved to be showing biological activity in the literature. So what we did is, so when we check the literature, simple this endogenous moieties were functionalized using transmetal catalysis, or maybe peroxide oxidant in presence of oxidant, or maybe under the influence of photocatalyst. So for our reaction condition, we chose this CFI. So CFI, I mean chromone fused endogenous CFI 1A, and treated with disulfide using different electrodes. So we tried carbon platinum, carbon nickel, carbon RBC, and different electrolytes and additives and solvents were screened, and we could find that I believe, yeah. So this NBO4 NPF6 found perfect electrolyte for this CS self-annihilation reaction in carbon as a anode and platinum as a cathode, and using Ki as a additive in acetonitrile solvent, we could isolate at 90% of this corresponding CFI thiethers 3AAA. Once we have optimized reaction condition in our hand, substrate scopes were studied. A library of these compounds were synthesized by reacting these different CFIs possessing electron donating and electron withdrawing group, and reacted well with the disulfide possessing neutral group and donating group and withdrawing group, as well as alkyl disulfide were also found compatible for this electrochemical CH sulfinylation reaction. Here there are few more examples. Further, we extended this electrochemical protocol for carbon selenium bond formation. I can say that CH selenylation reaction, and for which diselenides, variety of diselenides were charged with this CFI molecule 1 using graphite anode and platinum cathode, and this NeBF4 and PF6 as a electrolyte, Ki as additive at room temperature. We could obtain the corresponding seleno ethers, CFI containing seleno ethers in most of the cases in beyond 90% yield. These results we published in ChemCom very recently. So I am not going to show here the control experiments. So here also we performed the control experiment, but when we did the radical trapping experiments, we could not inhibit completely the reaction of this carbon sulfur bond formation. This suggested the two pathway of, there is a possibility of two pathway, that is one may be ionic pathway, other one may be radical pathway. I am not going detail of this reaction pathway. Yeah, we extended this protocol for the simple indologene moieties and the other cyclic ketone possessing indologenes, and you can see that in this case we could perform a CH mono chalcogenation, and in simple indologene we could perform the bis chalcogenation, and this result is under we just communicated, so I am not going in much detail of this project. No doubt, my group have collaboration with Professor Sharma from MLSU and Professor Lee from Taiwan. So in collaboration also we could develop some protocol like we could with Professor Sharma we could develop a electrochemical synthetic protocol for the synthesis of carbodiamides via oxidative cross coupling between amine and isocyanides, and we further extended this work for the synthesis of phenylphenidine under electrochemical reaction conditions. With Professor Lee, we could develop a facile protocol for the synthesis of aryl azophosphine oxides and phosphinoids via NP bond formation between amines and phosphorus surrogate using blue LED. And recently, we have also reported a cesium carbonate catalyzed synthesis of phosphanothiols via S-phosphonation of thiols under these reaction conditions. We could develop a synthetic protocol for the synthesis of 3H sulfinyl and selenyl 1H indoles by using 2-alkyne aniline as a starter precursor, and by reacting it with the disulfide and diselenide under the influence of potassium tetroxide to generate the desired products. Therefore, yesterday we have communicated this work where we just developed a palladium catalyzed synthetic protocol for the lauric indoles, that is 2,3 substituted indoles via the arylation of this alkynyl anilines with this silyl group. So this is the work in the collaboration with Professor Lee. So in conclusion, I can say that I could try to show you that we could develop my early 3 years. We could develop a transition metal free or sometime catalyst free protocol for carbon selenide and carbon phosphor bond formation. And from last almost 1.5 year, we are working on our core research scheme of, sponsored by SIR, where we propose electrochemical synthetic procedures, and by using electrochemical procedures, we could develop electrochemical reaction condition for the site selective CH sulfination, selenization, as well as cross coupling reaction. This is my laboratory. I have only laboratory. I don't have any instruments like NMR, mass, anything. I get it on charge basis from other institutes. Thanks to my students, they roam here to there to collect data. This is the ICA instruments where we performed all these electrochemical, electronic reactions. These are few pictures of my city, Pink City, Jaipur. I hope most of you might have visited Professor Chauhan sir, Prof. Kyan Singh sir, and I hope many of you might have visited. Again, I invite all of you to visit the beautiful Pink City. October to February is the best time to visit Jaipur. And this is Hawa Mahal. Jal Mahal. This is Hawa Mahal. This is the other view of Hawa Mahal, and this is Amer Fort. And with this, I come to the most important slide. I must thank all the funding agencies, UGC, DST for Inspire, SIR for YSS, as well as CRG, CSR for EMR. And this is my first batch of my students is Pratibha, she is Rakhi, she is assistant professor in Rajasthan, Bharpur. She is Rekha, she is doing postdoc. Kamlesh was a teacher candidate. He was working as a assistant professor in Rajasthan, college, Rajasthan RPC College. this is the present group of students. Whatever work I have shown for electroorganic synthesis was performed by Pooja, and some part was performed by Amreen and Kusum. He's Dr. Lalit. He will be moving very soon to Israel. Is Kamlesh over here when he was submitting the thesis. This is my colleague Asa. I thank my collaborator Professor Lee and Sharma, and finally once again thank you very much. I again thank the organizer of STIST 2023. Finally, thank you very much for kind attention and kind listening.
Any question? Any queries?
Yeah.
Yes, yes.
Yeah.
First of all, I would like to thank you for showing a special way of synthesizing organic molecule in electrochemistry. My question is, this is very good. We can apply electricity and produce a lot of molecules. Are there reactors already available in the industry to produce large quantity of drug molecules? So this way, it is possible. It is a potential way of synthesizing molecule, but are there already reactors available in industry?
Actually, very good question. The beauty of electroorganic synthesis is that you can play with the oxidation potential. We are all aware that any organic molecule have either oxidation potential or redox potential. And by changing the current or voltage, we can nicely play with the oxidation potential or redox potential. And due to lack of time, I could not go in much details. By changing the voltage, you can have formation of different products even, okay. So no doubt, this is what we have done so far at smaller scale, and I don't want to go in detail. We are in discussion with some pharma company, a leading pharma company of the country. They are asking us to, can we help them to establish, for gram scale synthesis. No doubt, this reaction we performed up to 3 millimole scale, okay. Because the problem with this ICA instrument is that the size of electrodes and the setup, okay. So if the setup can be, no doubt people are trying to use different sort of potentiometer to perform such kind of electrochemical synthesis. The problem with potentiometer is that you cannot have the constant current, because in the reaction media there will be formation of your products and other intermediates which will definitely change the resistance during course of the reaction, and which will create a trouble for the constant current or constant voltage. And that's the reason that ICA is able to sell a simple molecule up to cost of almost 4 lakh or 5 lakhs. So that's a major drawback, and people are trying. I hope in coming years things will be resolved. Thank you.
That will be the ultimate goal.
Yes, yes, yes. Thank you. Thank you.
Any other? Okay, if there are no more, may I call upon to hand over the memento to. There's a memento for you.
Yeah, sure. Thank you very much.
Thank you so much, Dr. Satpal. So that ends our first technical session, and I would like to call. I would like to call over the chairperson for this technical session, Professor PMS Chauhan, to come on the stage, please. I think they're going to hand over a memento of something. Come. Sir, please. You're coming. That's a Mizoram traditional stole. Thank you so much, sir. Thank you so much. So that ends our first technical session. The second technical session will start at 2, and in between, from now before 2, will be a lunch provided, and as well as a poster session going on outside. Thank you so much.